Welcome back to Bookfulness. This is our deep dive, episode 14.
Hi there. Yeah, last time we were really zoomed out, weren't we? Looking at those big evolutionary battles, species against species.
So evolution, yeah. Predators and prey, hosts and parasites,
Yeah.
all that dynamic external stuff.
Exactly. Very visible drama.
But today, we're doing the opposite. We're going micro, like really micro, all the way inside the cell into the nucleus.
Right into the blueprint itself. We're diving into chapter 14 today, focusing on how genes and genomes actually evolve.
So the mission today is, well, it's pretty fundamental, isn't it? Where does new genetic stuff come from? How do chromosomes get shaped? And maybe the biggest question, why do some organisms have just vastly more DNA than others?
That's the core of it. We want to understand the processes that create novelty, shape the large-scale structure, and, yeah, figure out why genome size is so weird.
Okay, let's start with weird. Because the first big hurdle, the thing you bump into right away, is this idea that genome size doesn't match up with complexity at all.
It's a classic, the C-value paradox. For ages, everyone just assumed, more complex organism, bigger genome. Makes intuitive sense, right?
Like a bigger machine needs a bigger instruction manual.
Exactly. But then you look at the actual data across life, and it just falls apart. You've got some species of amoeba single-celled organisms with, get this, up to 400 times more DNA than humans.
400 times. That's just wild. An amoeba.
It completely flips that intuition on its head. It tells you straight away that most of the DNA in that huge amoeba genome, and frankly, a lot of the DNA in our genome, isn't coding for complexity in the way we usually think about it.
So we have to rethink what important DNA even means.
Absolutely. And just a quick note for everyone listening, we'll be mentioning figures from the chapter, like figures 14.1 and 14.2, which really show the size disparity. You can find all those figures in the file linked in the description. Definitely worth a look.
Great. Okay, so let's park the why so much DNA question for a bit and start with the parts we generally do think of as important, the genes themselves. Where do new ones actually come from?
Right. Before we get to the bulk, let's stick with the genes for a moment. If total DNA amount, the C-value, doesn't correlate with complexity, maybe the number of protein-coding genes does.
You'd think so, wouldn't you? But again, the numbers are, well, humbling. Humans, Homo sapiens, we have about 20,000 protein-coding genes, give or take.
And the surprising bit, that tiny little roundworm C. elegans, it has roughly the same number, maybe 19,000, 20,000.
A microscopic worm versus a human, built with basically the same number of core genetic parts. That's something.
It really is. And if you want the champion, the organism with the most known genes, it's not us. It's not a whale. It's breadwheat.
Breadwheat. Seriously.
Yep. Common breadwheat has something like 100,000 genes. 5 times as many as we do.
Okay, so complexity clearly isn't just about the number of genes either. It must be about how they're used, how they're regulated.
Precisely. Regulation, expression patterns, alternative splicing, we'll get into that. It's the network, not just the nodes. So, yeah, most of the size difference we see across genomes is down to the non-coding DNA. We'll definitely circle back to that.
But for now, let's focus on creating the building blocks. If you need a new function, a new adaptation, you generally need new or modified genes. What's the main way evolution makes them?
The number one source, especially in eukaryotes like us, is gene duplication. It sounds simple, but it's incredibly powerful.
Just making extra copy.
Basically, yeah. Usually it happens because of errors during DNA replication. The cellular machinery messes up, copies a chunk of DNA twice. Sometimes that chunk contains a whole gene, sometimes just part of one, sometimes several.
And the key thing is the original gene is still there doing its job.
Exactly. That's the evolutionary magic trick. If you only have one copy of a really critical gene, say something essential for basic metabolism, any mutation could be bad news, maybe even lethal. Selection keeps it locked down tight.
But if you suddenly have a spare copy,
then that spare copy is, initially at least, redundant. It's not under the same intense pressure. It's free to accumulate mutations, to drift, to potentially stumble upon a new function without immediately breaking anything vital. The original copy is the safety net.
so duplication creates raw material by creating redundancy.
That's the heart of it. Redundancy equals opportunity. And while single gene duplications are happening all the time, driving a lot of finer scale evolution, we should also mention whole genome duplication, or WGD.
Copying the entire set of chromosomes.
Yeah. It's much rarer, obviously a huge event, but it's been incredibly important in some lineages, especially in plants. It seems to have fueled massive bursts of diversification. Imagine suddenly having 2 copies of every gene. That's a colossal amount of raw material to work with.
Okay, let's make this concrete. Is there a good human example showing this duplication process happening like relatively recently and driven by selection?
Absolutely. The AMY1 gene is perfect. It codes for salivary amylase.
The enzyme in your spit that starts breaking down starch.
That's the one. And what's fascinating is that humans show copy number variation CNV for this gene. Different people and different populations have different numbers of copies of AMY1.
And this isn't just random variation.
Not at all. It correlates beautifully with diet. The more copies of AMY1 you have, the more amylase you produce in your saliva, and the better you are digesting starch right from the first bite.
So you'd expect populations with historically starchy diets.
To have more copies.
Yeah.
And that's exactly what the data shows. If you look at populations with a long history of agriculture, relying heavily on things like rice, wheat, potatoes, think Japanese populations, European Americans, they tend to have, on average, maybe 6 to 8 copies of AMY1.
Okay, and what about populations with different diets?
Contrast that with groups that traditionally relied much less on starch, like hunter-gatherer populations. The Biaka people in Central Africa are often cited their average copy number is lower, maybe 4 or 5 copies typically.
Wow. So easily a 50% or more difference in average copy number directly linked to the selective pressure of diet. That's recent human evolution in action.
It really is. The selective advantage of being able to efficiently digest abundant starch favored individuals who, by chance duplication, happen to have more AMY1 copies. Those copies were retained and spread. It's a clear signal of diet-driven adaptation working on gene duplication within, historical timescales.
And this isn't a one-off. The genome is constantly changing like this.
Constantly. Gene duplication and gene loss are continuous processes. If you compare the genomes of related mammals, humans, chimps, mice, dogs, you find evidence for thousands of gene families being gained or lost just within those lineages over, relatively short evolutionary periods. It's a dynamic landscape. Genes appear, genes disappear, copy numbers shift. The genome is definitely not static.
Okay, so duplication is the big one for generating new gene material. But you mentioned the genome is creative. Are there other ways evolution cobbles together new genes?
Yeah, duplication is the main engine, but there are a couple of other really neat mechanisms. Think of them as secondary routes to novelty.
Like what?
Well, first there's exon shuffling. Remember how eukaryotic genes are often broken up into coding bits, the exons, separated by non-coding introns?
Right, that modular structure.
That structure is key. Exon shuffling is basically taking existing functional exons, often coding for specific protein domains, like a bit that binds DNA or a bit that forms a channel, and rearranging them, stitching them together in new combinations.
So instead of inventing a whole new function from scratch through random mutation, you're mixing and matching pre-approved parts,
Yeah,
like genomic Lego.
that's a great way to think about it. You take a working module from gene A, maybe another from gene B, and stick them together to create a new gene, gene C, with potentially a novel combined function. It can dramatically speed up the evolution of complex proteins.
Is there an example of this?
The book mentions the Jingwei gene in Drosophila fruit flies. It seems to have formed when an exon from one gene got fused onto a duplicated copy of another gene. The resulting hybrid gene has a new structure and function derived from this shuffling event.
Cool. Okay. What's the other secondary source?
Horizontal gene transfer, or HGT. This is when genetic material moves between organisms that aren't directly related through reproduction. Think bacteria swapping genes.
Right, not parent to offspring, but sideways, between different species or lineages.
Exactly. It's super common and incredibly important in prokaryotes like bacteria and archaea. Less common in complex eukaryotes like us, though it does happen, sometimes via viruses.
Why is it so important for bacteria?
Because it allows for adaptation in a fell swoop, as the book puts it. A bacterium doesn't have to wait to slowly evolve resistance to an antibiotic through gradual mutations. It can just acquire a fully functional resistance gene directly from another bacterium that already has it, maybe via a plasmid.
Ah, so that's why antibiotic resistance spreads so terrifyingly fast. It's HGT.
It's a huge factor, yes. Bacteria are constantly sharing useful genes horizontally, which allows populations to adapt very, very quickly to new challenges like antibiotics or different food sources.
Okay, so duplication is primary. Shuffling and HGT are secondary ways to get new genetic material. Let's go back to duplication, the main source. Once you have that spare copy, what happens to it? Does it always lead to a new function?
Not at all. That spare copy faces several possible outcomes. The book outlines 4 fates of a duplicated gene, and the fate depends on selection pressures, or the lack thereof.
Okay, fate number 1.
Loss. This is actually the most common fate. The duplicated copy is often just extra baggage. If there's no immediate benefit to having more of the protein, random mutations will start to accumulate in that spare copy.
Because it's not under selection anymore.
Right. Eventually, one of those mutations will likely break it, introduce a stop codon, mess up the reading frame, becomes non-functional. We call this a pseudogene. It's still there in the DNA, like a fossil, but it doesn't make a working protein, so most duplicates just fade away.
Okay, common fate. Fizzle out. What's fate number 2?
Retention. This is simpler. Sometimes just having more of the original protein is beneficial. The dose makes the difference.
Like our AMY1 amylase example. More copies, more enzyme, better starch digestion.
Exactly like AMY1. The extra copy is kept simply because increased expression, making more product, provides a selective advantage. It's optimization through quantity.
Straightforward. Fate number 3.
This is the exciting one. Neofunctionalization. Neo, meaning new. This is when the duplicated gene accumulates mutations that give it a completely new function, one different from its ancestor.
This is where true innovation comes from.
Yes, this is how evolution really explores new possibilities. The duplicated gene, free from its original constraints, wanders off into a new functional territory.
And the electric fish example is a classic case of this, right?
Yeah.
Showing it happened independently.
It's a fantastic example. Electric organs evolved separately in South American knife fishes and African elephant-nosed fishes. Different lineages, same solution. And how did they do it?
Duplication.
Duplication of a gene for a sodium channel protein, N4A. The original gene, let's call it SHIN4A, is still doing its normal job in muscle cells, triggering contractions.
But the duplicate.
The duplicate, SHIN4A, underwent neofunctionalization. Mutations accumulated that caused it to stop being expressed in regular muscle but start being expressed at high levels in specialized cells called electrocytes, which make up the electric organ.
So it repurposed the sodium channel mechanism from making muscles twitch to generating electric field.
Precisely. Same basic molecular tool, completely new application for hunting, for sensing, for communication in murky water. And the fact that a similar duplication then neofunctionalization event happened independently in 2 different groups highlights how powerful this pathway is.
Amazing. Okay, that leaves fate number 4.
Subfunctionalization. This one is a bit more subtle. It happens when the original ancestral gene actually had multiple jobs, or was active in multiple tissues, or at multiple times.
Like a multitasking gene.
Right. After duplication, there are now 2 copies doing the same multiple jobs. Selection might then favor mutations that specialize each copy. One copy loses function A but keeps function B, while the other copy loses function B but keeps function A.
So they divide the labor. The original gene's tasks get partitioned between the 2 duplicates.
Exactly. Each duplicate becomes specialized for a subset of the ancestral functions. A really clear example is fetal hemoglobin in mammals.
How does that work?
Our adult hemoglobin is great at carrying oxygen, but fetal development poses a challenge. The fetus needs to get oxygen from the mother's blood across the placenta.
And the mother's hemoglobin isn't keen to just give it up.
Right. So what evolved? An ancestral globin gene duplicated. One copy remained the adult version. The other copy, through subfunctionalization, evolved to be expressed only during fetal development and, crucially, mutated to have a higher affinity for oxygen than the adult form.
Ah, so the fetal hemoglobin can effectively pull oxygen away from the maternal hemoglobin in the placenta.
Exactly. It outcompetes it. This specialization, enabled by duplication and subfunctionalization, is essential for mammalian development. The 2 genes divided the temporal labor, one for fetal life, one for postnatal life, and optimized for each stage.
Loss, retention, new function, split function, those are the fates.
Yeah.
And you also mentioned genes can physically move. Gene trafficking.
Yeah, it's another layer of dynamism. Genes aren't necessarily stuck in one place on a chromosome. They can move, sometimes through mechanisms involving transposable elements, which we'll get to. This can change how they're regulated because the genomic neighborhood matters. The book notes a pattern in Drosophila where genes seem to have preferentially moved off the X chromosome onto the autosomes, the non-sex chromosomes. Why exactly is complex, but it shows the genome map itself is fluid over evolutionary time.
Okay, so we have these processes, duplication, shuffling, HGT generating variation, and then these different fates playing out. But how do we actually tell when we compare gene sequences between species whether the differences we see are the result of adaptation, positive selection, finding a better version, or just random genetic drift, accidents that stuck?
That's the million-dollar question in molecular evolution, and the framework for answering it really comes from Motu Kimura and his neutral theory of molecular evolution, which he proposed back in the late
The
60s.
neutral theory. I feel like this is fundamental.
It absolutely is. Kimura's big counterintuitive idea was that most of the genetic differences you see accumulating between species over time at the molecular level are not due to positive natural selection fixing beneficial mutations. Instead, he argued, most of them are due to random genetic drift fixing mutations that are effectively neutral or maybe very slightly harmful, but not harmful enough for selection to weed them out efficiently.
So most molecular change is just noise, evolutionary noise.
In a sense, yes. Or rather, it provides the essential baseline against which we can detect the signal of selection. The theory starts by thinking about the types of mutations.
What?
You have non-synonymous mutations. These change the amino acid sequence of the protein. Since proteins are usually pretty well tuned for their jobs, changing an amino acid is often bad. It might break the protein's structure or function.
So selection gets rid of those.
Exactly. That's purifying selection. It purges deleterious mutations, keeping the protein sequence stable over time.
Okay, and the other type?
Synonymous mutations. These change the DNA sequence, but because of the redundancy in the genetic code, they don't change the resulting amino acid. The protein stays the same.
So these should be invisible to selection.
Largely, yes. They're considered effectively neutral. Their fate, whether they disappear or spread through the population and eventually become fixed, is determined mostly by random chance, by genetic drift. And they should accumulate at a relatively steady rate, like a molecular clock.
Okay, so purifying selection acts strongly against non-synonymous changes, while synonymous changes drift neutrally. What does that predict?
It predicts that, for most genes under typical functional constraints, the rate of synonymous substitutions, let's call it sexy SEL, should be higher than the rate of non-synonymous substitutions, because the harmful day-dollar changes are constantly being removed by purifying selection, while the neutral day-dollar changes just pile up due to drift.
And that prediction gives us a tool.
It gives us the key tool, the D-dollars axio. You calculate the rate of non-synonymous changes between two sequences and divide it by the rate of synonymous changes. This ratio tells you what kind of selective pressure has been operating on that gene.
Right, let's break down the possible outcomes. What does DNDS S and Ns mean?
D-dollars S81 is the most common result. It means synonymous changes are happening faster than non-synonymous ones. This is the signature of purifying selection. The gene is important, and evolution is actively preventing changes to the protein sequence. This fits the neutral theory's expectation for a functional gene.
Okay, what if the ratio is equal to 1?
DNDS equals day-lun suggests neutral evolution. Non-synonymous changes are accumulating at a same rate as synonymous ones. This implies there's no real selective pressure on the protein sequence. Changes are just drifting. You might see this in pseudogenes, for example, or perhaps genes with very relaxed constraints.
And the really interesting one, DNDS greater than 1?
DNDS 1-1. This is the smoking gun for positive selection. It means that non-synonymous changes to the protein have been fixed by selection faster than neutral mutations have accumulated by drift. This only happens when evolution is actively favoring changes to the protein sequence, driving adaptive evolution at that specific gene. It's a relatively rare signal, but a very strong one when you find it.
Because D-Lawrence is always ticking along in the background due to drift, so D-Lawrence has to be accumulating really fast to overtake.
Exactly. You need strong, recent positive selection to push that ratio significantly above 1.
The book uses the Duke-Langor monkey as an example of this positive selection signature.
Yes, it's a great case study. Duke-Langors are leaf-eating monkeys, and they have a specialized foregut fermentation system, kind of like a cow. They rely on bacteria to break down cellulose.
And they needed an enzyme to deal with all those bacteria.
Right. They needed an effective way to digest the bacteria themselves, to reclaim the nutrients. They duplicated a gene for an enzyme called RNase 1, which breaks down RNA. One copy, RNA SE1B, was repurposed for this digestive role in the gut.
And when they looked at the evolution of that specific copy,
They found a very clear signal of positive selection. In the roughly 4 million years since that gene arose via duplication, there were 9 non-synonymous substitutions fixed, compared to only 3 synonymous ones. That gives a DN-DSI ratio significantly greater than 1.
So selection was rapidly tweaking the enzyme sequence to make it better at its new job of digesting bacteria in the gut environment.
Precisely. It's a textbook example of using the DNDS ratio to pinpoint adaptive evolution at the molecular level.
Okay, so we can detect positive selection. But going back to Kimura's idea, how much of overall protein evolution is actually adaptive versus just drift? Does it vary?
It varies hugely. In organisms with really large population sizes and fast generation times, think fruit flies, bacteria selection is much more efficient. Even slightly beneficial mutations have a good chance of being picked up. So in Drosophila, some studies estimate that maybe up to 50% of the amino acid differences between species might be adaptive, fixed by positive selection.
But what about us? Humans, mammals?
Our effective population sizes are much smaller, which means genetic drift is a much stronger force relative to selection. So while positive selection definitely happens on specific genes where the pressure is strong, like AMY1, a lot more of the background protein evolution in mammals seems to be driven by drift. The estimate cited is that maybe less than 15% of recent protein evolution in the human lineage is adaptive.
So drift plays a bigger role in shaping our proteome overall, even though selection is critical for specific adaptations.
That seems to be the picture, yeah.
One last point in this section. We said synonymous sites are usually neutral, but are they always perfectly neutral?
Good question. Mostly, but not entirely. There's a phenomenon called codon bias.
Meaning?
The genetic code is redundant, right?
Yeah.
Several different 3-base codons can specify the same amino acid. But if you look within a genome, these different synonymous codons are often used at very different frequencies. Some codons for, say, leucine might be much more common than others. That's codon bias.
Why would there be a bias if they all make the same amino acid?
It often comes down to translational efficiency. The cell has a pool of transfer RNA, tRNA molecules, each recognizing a specific codon. If the cell needs to make a lot of a particular protein very quickly, selection can favor using the codons whose corresponding tRNAs are most abundant or efficient.
So using the fast lane codons speeds up protein production.
Exactly. It provides a small selective advantage, especially for highly expressed genes. This selection for translational efficiency is strongest in organisms with huge population sizes, like microbes, where even tiny fitness differences matter. In organisms like us, with smaller populations, drift tends to overwhelm this weak selection, and codon bias is much less pronounced. So synonymous sites aren't perfectly neutral, but the selection on them is usually pretty subtle compared to selection on non-synonymous sites.
Okay, we've spent a lot of time on changes within the coding sequence, the DNDS story. But you hinted earlier that sometimes the big evolutionary action isn't changing the protein itself, but changing how it's used.
Absolutely. Evolution of gene expression, changing when, where, or how much a gene is turned on or off is arguably just as important, if not more so sometimes, than changing the protein sequence.
Tweaking the regulation rather than the hardware.
Precisely. A small change in a regulatory element, like a promoter or enhancer sequence upstream of a gene, can have huge downstream effects on the organism's phenotype without ever touching the protein code.
Is there a good example of adaptation happening this way?
The mosquito Aedes aegypti is a really compelling case. This species is a major vector for diseases like Zika, dengue, yellow fever. And one key aspect of its evolution is its shift towards preferentially biting humans.
How did it adapt to target us specifically?
Part of the story involves a gene called OR4, which codes for an odorant receptor protein that helps the mosquito smell its potential hosts. Research found that populations of Aedes aegypti that strongly prefer humans didn't necessarily have changes in the OR4 protein itself. Instead, they had mutations in the regulatory region controlling the OR4 gene.
And these mutations changed.
They caused the OR4 gene to be expressed at much higher levels. Simply turning up the volume on this one receptor made the mosquitoes significantly more sensitive or attracted to chemicals specific to human scent.
Wow. So a regulatory tweak helps drive a major ecological shift specializing on humans. That's powerful.
It really highlights how crucial regulatory evolution is. Adapting to a new environment or lifestyle might often involve tinkering with the existing gene toolkit's expression patterns rather than inventing entirely new tools.
Okay, that connects back to gene structure too. You mentioned introns and exons earlier. Let's revisit that. Eukaryotic genes have these coding exons separated by non-coding introns.
Yes, that split structure is fundamental to eukaryotes.
And it enables another layer of complexity, right? Alternative splicing.
Exactly. When the gene is transcribed into RNA, the introns have to be spliced out and the exons stitched together to make the final messenger RNA, mRNA, that gets translated into protein. But the cell doesn't always have to stitch all the exons together in the same way.
It can pick and choose which exons to include.
Precisely. It can skip certain exons or use different splice sites. This means that from a single gene sequence in the DNA, a cell can generate multiple different mRNA molecules and therefore multiple different protein variants or isoforms.
So one gene can actually code for many related but distinct proteins.
Yes, potentially dozens or even hundreds from a single gene locus. It's a major way eukaryotes generate protein diversity and functional complexity without needing an absurdly huge number of genes. It's like having a recipe book where you can mix and match paragraphs to create different dishes.
This helps explain the C-value and gene count paradox we started with. Humans have only 20,000 genes, but we make vastly more proteins than that. Alternative splicing is a key part of the solution.
It's a huge contributor, absolutely. It dramatically expands the functional output of the genome.
Okay, so we have coding exons, regulatory regions, introns, enabling alternative splicing. Let's put it all together and look at the big picture again. What is the overall composition of the human genome? That figure you mentioned earlier.
Right, figure 14.14 in the book. It's quite an eye-opener if you break down the entire 3.2 billion base pairs of the human genome.
The actual protein coding exons make up how much?
Less than 2%. Maybe 1.5%.
1.5% codes for all the proteins in our bodies. What's the rest?
Well, a big chunk, maybe 25–30%, is introns and regulatory sequences associated with those genes. But the really dominant component,
Is the stuff we talked about potentially being junk?
that's where transposable elements, TEs, come in.
Okay.
They make up the largest single fraction of the human genome. Estimates vary slightly, but it's somewhere around 45–50%.
Almost half of our DNA consists of these mobile genetic elements or their remnants.
That's right. So less than 2% protein coding, maybe 25% introns and regulatory bits, and nearly 50% transposable elements. The rest is other kinds of non-coding sequences, repeats, etc. It paints a very different picture than just a lean, mean coding machine. Our genome is vast, and most of it isn't genes in the traditional sense. Which brings us nicely to chromosome evolution and the question of all that other DNA.
Okay, so we've gone from single genes to the whole genome composition. Now let's zoom back out a bit, but stay inside the nucleus. Let's talk about the chromosomes themselves, these large structures that package the DNA. How does their number and structure evolve?
Right, the karyotype, the characteristic chromosome complement of a species. It can change quite dramatically over evolutionary time. The main ways the number changes are through chromosome fusions, where two chromosomes join end-to-end to become one larger chromosome, and fissions, where one chromosome breaks into two smaller ones.
We see different chromosome numbers across species, like humans have 46, 23 pairs, but chimps have 48. That difference likely arose from a fusion event in our lineage after we split from chimps.
Exactly. Human chromosome 2 looks very much like it's the result of fusing two ancestral ape chromosomes. You can even see the remnants of the telomeres, chromosome ends, and an extra centromere in the middle.
So these fusion and fission events happen. But why do they happen, and why do they sometimes spread through a population? Is it always because having, say, 46 chromosomes is somehow better than having 48? Is it adaptive selection on the organism?
That's the intuitive assumption, but the reality seems much weirder and more fascinating. Often, changes in chromosome number aren't driven by selection for the survival or reproduction of the organism, but by conflicts happening within the process of making sperm and eggs itself. This is called meiotic drive.
Internal conflict.
Right.
Selfish genes at the chromosome level.
Precisely. Think about meiosis, the cell division that produces gametes, sperm and eggs. In females, only one of the four resulting cells becomes the egg. The other three become non-functional polar bodies and degenerate. Meiotic drive occurs when a particular chromosome or gene variant somehow cheats during this process to ensure it gets into the egg more often than the 50% chance expected by Mendelian inheritance.
It's rigging the lottery.
Kind of, yeah. The book uses a great example from house mice, figure 14.15. In some populations, there's variation in whether certain chromosomes are fused together, metacentric, or remain separate, acrocentric. And it turns out, during female meiosis, the unfused acrocentric chromosomes have an advantage. They preferentially orient themselves on the meiotic spindle in a way that makes them more likely to end up in the egg nucleus, while the fused metacentric versions are more likely to be shunted off into the polar bodies.
So the unfused chromosomes are essentially elbowing their way into the next generation.
Exactly. It's a selfish advantage at the level of chromosome transmission during meiosis. This drive can cause the unfused condition to spread rapidly through the population, changing the overall karyotype, even if having more smaller chromosomes isn't necessarily better for the mouse's overall survival or health.
That's incredible. It means the chromosome number we see isn't necessarily an optimal adaptation, but could just be the messy outcome, as the book says, of these internal genetic conflicts playing out over evolutionary time.
It really challenges the idea of the genome as a perfectly harmonious system. It's often a battleground for competing genetic elements.
Okay, besides number changes like fusions and fissions, what about changes to the structure within a chromosome?
A really important type of structural change is a chromosome inversion. This is when a segment of a chromosome gets flipped end to end. The genes are all still there, just in a reversed order within that segment.
Doesn't sound too dramatic if all the genes are still present. Why are inversions evolutionarily significant?
The key property is that inversions suppress recombination.
How does flipping a segment stop recombination?
Think about an individual who is heterozygous for the inversion. They have one normal chromosome and one chromosome with the inverted segment. During meiosis, these two chromosomes try to pair up gene for gene. To do that, the inverted region has to form a physical loop to align correctly with the normal sequence.
Okay, I can picture that loop.
Now, if a crossover event, recombination, happens within that loop, the resulting chromosomes get messed up. You can end up with chromosomes that have duplications, deletions, 2 centromeres, or no centromere. These are often unbalanced and lead to non-viable sperm or eggs.
So effectively, successful recombination within the inverted region is prevented in heterozygotes.
Right. The genes inside the inversion tend to get inherited together as a single block, almost like a supergene.
And that's the evolutionary advantage, locking alleles together.
Yes. If you have a set of alleles at different genes within that inverted region that work particularly well together, a co-adapted gene complex, the inversion protects that winning combination from being broken up by recombination in heterozygotes. It keeps the successful team together.
Can we see this in action?
The ruff, a bird known for its incredibly diverse male mating strategies, is a prime example. Figure 14.1 summer. There are territorial males, independents, smaller satellite males that associate with them, and even rarer fader males that mimic females.
And these complex differences in appearance and behavior?
Are largely controlled by different versions of a large chromosomal inversion. The specific alleles for plumage, size, and mating tactics that define each morph are locked together within different arrangements of this inversion. The inversion ensures that the whole suite of traits for being, say, a satellite male gets passed down as a coherent package deal.
All right, let's bring it all home. We started with the C-value paradox. Why do genome sizes vary so wildly and seemingly independently of complexity? We established it's mostly about non-coding DNA. Now let's really dig into why that non-coding DNA accumulates.
Okay, revisiting that puzzle, figure 14.rhent clearly shows this trend. Prokaryotes, like bacteria, have incredibly streamlined genomes, almost all coding DNA. Very little space wasted. They're lean and mean.
Efficiency seems key for them.
Definitely. But as you move to eukaryotes, especially multicellular ones like animals and plants, the proportion of coding DNA drops dramatically, and the total genome size can explode. And we identified the main culprit for that explosion.
Transposable elements,
Uh-huh.
TED. The junk DNA, or maybe more accurately, the selfish DNA.
Exactly. TEDs are the dominant component of these large eukaryotic genomes. They are essentially genetic parasites. Their evolutionary success isn't measured by how much they benefit the host organism. They often don't, or are even harmful. Their success is measured purely by how good they are at making copies of themselves and inserting those copies elsewhere in the genome.
Like a virus, but one that lives permanently within our own DNA and just focuses on replicating itself there.
That's a very good way to put it. They hijack the host cell's machinery to proliferate. They exist because selection acts on them, favoring variants that are better at copying and pasting themselves.
You mentioned the ALU element in humans. How common is that?
Incredible common. It's a short sequence, only about 300 base pairs. But there are over 1 million copies of ALU scattered throughout your genome right now. Add them all up, and they constitute more than 10% of all your DNA.
10%, just from this one type of element. How does it copy itself?
ALU is a type of retrotransposition. It works via an RNA intermediate. The ALU DNA sequence gets transcribed into RNA. Then, critically, an enzyme called reverse transcriptase makes a DNA copy from that RNA template, and then that new DNA copy gets inserted somewhere else in the genome.
Where does it get the reverse transcriptase? Does it make its own?
That's the sneaky part. ALU doesn't even have its own gene for reverse transcriptase. It's what's called a non-autonomous retrotransposon. It relies on borrowing the reverse transcriptase enzyme produced by other active transposable elements, like line elements, that do encode their own machinery. It's a hyperparasite, mooching off the tools of other parasites.
Wow, efficient parasitism. Can we trace the history of these things in our genome? When were they most active?
Yes, we can estimate the age of different TE copies by looking at how much they've mutated since they first inserted. When you do this for the human genome, figure 14.19 shows this history, you see that TE activity isn't constant.
What's the pattern for us?
Right now, thankfully, TE activity in humans is relatively low. Our genomes have evolved sophisticated defense mechanisms, mostly epigenetic silencing like DNA methylation, to shut down most active TEs. But the analysis reveals a massive burst of TE activity in our primate ancestors, particularly the ALU element peaking somewhere around 40 to 50 million years ago.
So we're living with the ghosts of an ancient genomic invasion.
In a very real sense, yes. Our large genome size is, in large part, the accumulated debris from that period of intense TE proliferation millions of years ago.
These things sound mostly bad. What kind of damage do TEs cause?
Their main negative impacts are mutational. Firstly, if a TE inserts itself right into the middle of a functional gene or critical regulatory sequence, it can completely disrupt its function. That's insertional mutagenesis.
Okay, direct damage.
Secondly, having hundreds of thousands or millions of nearly identical sequences scattered throughout the genome creates hotspots for problems during meiosis. If the recombination machinery mistakenly pairs up two different ALU copies on sister chromatids or homologous chromosomes, it can lead to unequal crossing over, resulting in dilutions or duplications of large chromosomal segments. These structural rearrangements are often highly deleterious.
So the genome is constantly fighting back, trying to silence them.
Absolutely. There's an ongoing coevolutionary arms race between Ts trying to replicate and the host genome evolving defenses like methylation and RNA interference pathways like piRNAs, specifically to find and shut down TE activity.
But is there any upside? Can these parasites ever accidentally do something good?
Occasionally, yes. By random chance, a TE insertion might create a beneficial mutation. The classic example mentioned in the book is the industrial melanism in the peppered moth, Biston betullaria.
The moths that turned black during the Industrial Revolution.
That's the one. The mutation causing the dark coloration, which provided camouflage on soot-covered trees, was found to be caused by a large TE inserting into a regulatory region near a gene called cortex, changing its expression pattern during wing development. A TE insertion literally drove a rapid, famous adaptation.
And you mentioned earlier the even more profound idea that TEs might be the origin of introns.
Right, that's a major hypothesis. Perhaps introns started as selfish TEs that inserted into genes. The host cell then had to evolve splicing machinery just to remove these unwanted interruptions to maintain the protein code. Over time, this splicing machinery became more sophisticated, and the presence of introns, originally a nuisance, was co-opted to allow for alternative splicing, vastly increasing the genome's functional capacity.
Turning a bug into a feature on a grand evolutionary scale.
If true, it's one of the most remarkable examples of evolutionary co-option imaginable, making a core component of eukaryotic complexity out of a molecular parasite.
Okay, so wrapping it all up. Why are some genomes huge and full of TEs, while others stay small and lean? What determines the equilibrium size?
It seems to boil down to a balance between the rate at which TEs proliferate and the strength of selection acting against genome size increase.
So bacteria.
Selection for replication speed and metabolic efficiency is incredibly strong. There's intense pressure to keep the genome small and compact. Any unnecessary DNA, including TEs, is rapidly purged.
But in organisms like vertebrates,
We tend to have much smaller effective population sizes. This means genetic drift is more powerful and selection is less efficient. Specifically, selection against slightly deleterious mutations like the metabolic cost of replicating extra DNA or the slightly increased risk of harmful rearrangements caused by TEs is weaker.
so TEs can accumulate more easily because the host's ability to fight back via selection is weaker.
Essentially, yes. The TE's inherent drive to proliferate can overwhelm the relatively weak selection pressure, favoring smaller genomes in organisms with small population sizes. So our large TE-laden genomes might be less about adaptive benefit and more about the victory of selfish DNA in a context where selection couldn't effectively stop it. #tag outro. So wrapping up this deep dive, we've journeyed through the genome's, well, really quite messy evolutionary history, haven't we? We saw how new genes, new functions, primarily bubble up from the redundancy created by gene duplication accidents becoming opportunities.
And we learned we can actually measure the footprint of adaptation at the molecular level using tools like the DNDSI ratio, separating that positive selection signal from the constant background hum of genetic drift.
We also confronted the sheer scale of non-coding DNA, realizing that much of our genome's bulk comes from the history of transposable elements, these genetic parasites shaping genome size. And even fundamental features like chromosome number aren't necessarily optimized designs, but often the outcome of internal conflicts like meiotic drive.
Yeah, the big takeaway for me is just how much the genome isn't like a perfectly engineered blueprint. It's much more like I don't know, a rambling historical document full of crossings out, pasted in notes, old battle scars from internal arms races between selfish elements and host defenses. It's a product of its chaotic history.
That's a great way to put it. And maybe the most provocative thought, stemming from that meiotic drive example, is realizing that the very structure and number of our chromosomes, things we think of as bedrock biology, might just be the frozen accidents of ancient molecular cheating during reproduction.
Which definitely makes you wonder, if something as basic as chromosome number can be shaped by internal conflict rather than organismal benefit, what else about our biology might be like that? What other features that we assume are finely tuned adaptations might actually be, non-optimal compromises left over from evolutionary conflicts we don't even fully understand yet?
It opens up a whole new perspective, doesn't it? Looking for the history of conflict,
Yeah.
not just the story of adaptation.
Absolutely. Well, next time, we're going to take these genetic parts and connect them much more directly to the whole organism's form. We'll be diving into the fascinating field of evo-devo evolutionary developmental biology.
Yeah, we'll explore how changes in a surprisingly small shared toolkit of developmental genes can generate the incredible diversity of animal body plans we see around us. Should be really interesting.
Looking forward to it. Thanks for listening to Bookfulness.
Join us next time.